Welcome back to part 5 of our COVID-19 literature updates. Today we are going to be covering a wide variety of subjects and questions based on studies that have become available over a 13-day period from May 6th to the 18th. Once again, I am Aaron Richterman, the HIV Fellow at Brigham and Women's Hospital, and my co-presenter is Eric Meyerowitz, the HIV Fellow at Massachusetts General Hospital. Next slide. We continue to see a huge number of papers and preprints coming out, and as time goes on, we are seeing a larger and larger number of high-quality studies that are informing our understanding of this new disease. Next slide. Here's our agenda for today. We're going to start with a new study on racism and COVID-19 in the United States, discuss some new evidence describing the overall number of infections, go through new data on the origin story of SARS-CoV-2, walk through a few new important studies of antibodies, immunity, and vaccines, offer an answer to the question of whether there's a role for PCR testing in later disease. We'll highlight the concerning new inflammatory syndrome described in children that may be related to COVID-19. We'll walk through some large observational data on real-time symptom monitoring to predict disease that is coming out of mobile apps. Discuss the theoretical role of BCG along with some recent observational data on this subject, and finally update a few new treatment trials. Next slide. Okay, next slide. So bringing together a number of the points we tried to make during our last presentation, this preprint posted on May 11th is a cross-sectional study of publicly reported COVID-19 mortality data, attempting to systematically assess the quality of race ethnicity data in the United States and to estimate age-adjusted disparities using random effects meta-analysis. So they found only 28 states and New York City reported race ethnicity stratified COVID-19 mortality, as well as large variations in missingness by state, as you can see in the heat map on the left, with those in gray having no missing data, and the lighter blue having quite a bit of missing data. And so you can see on the four spots on the right, the top one being the overall risk, relative risk of death for Black people. And they found that an overall age-adjusted risk of 3.57 compared to white people. And in the four spot below, they found a relative risk for Latinx people of 1.88. They note that the I-square was greater than 90% in both analyses, indicating substantial heterogeneity between the states, perhaps somewhat related to missingness, as they found in meta-regression analysis significantly lower inequities in states with a higher proportion of deaths with missing race data. This is an incredibly important topic that we plan to continue following closely. Next slide. And since the time of the initial epidemic in China, there have been fierce debates as to the actual number of infections occurring, given that testing was being done on some subset of the total infected population, with a bias towards the more severe or hospitalized cases. Early reports from the WHO suggested that there were almost no asymptomatic or identified cases, such that the reported number of cases in China closely approximated the actual number of infections. Since then, however, we've seen a substantial amount of evidence of unidentified and to some extent asymptomatic disease, much of which we've covered in our prior presentations. So in thinking about this, testing identifies the tip of the iceberg, and understanding how big the iceberg is would be incredibly informative to our understanding of the aggregate severity of infection and is likely to have implications for population immunity. Next slide. 4 weeks ago, we discussed this preprint from a Stanford group posted on April 17th. That was the first attempt at assessing population seroprevalence conducted in Santa Clara, California. The methodology has been much criticized in terms of both sampling bias and their prevalence calculations. But the major takeaway, I think, is that this study was the first suggestion that while prevalence of infection is likely to be a good deal higher than the number of identified cases, it still remained fairly low in the 0 to 6% range. Next slide. Since then, we've seen additional serosurveys in varying populations and with varying sampling methods. New York State, which is probably some of the highest rates of disease in the world, conducted a statewide serosurvey over a 2-week period, performing serology testing on 15,103 people across the state who were recruited in grocery stores and community centers. We should note well that this is thus not a completely random or entirely representative sample, as those who find themselves in grocery stores or community centers during a pandemic may very well be unlike the general population in important ways. That said, after population weighting and correcting for testing characteristics, they found an overall prevalence of 12.3% in New York State on May 1st. If we consider that there were just over 300,000 reported cases as of May 1st, and ignoring for the moment adjustment for delay to seroconversion, this does suggest a rough approximation of 10 infections per identified case. Not as high as we might have hoped, but in line with what many experts have predicted in terms of being about one order of magnitude difference. They also reported difference in prevalence by region, and you can see that New York City had close to a 20% seroprevalence as compared to much lower rates in some other areas of New York State, as seen below. Again, as an example, and as a very rough estimate, with close to 14,000 deaths from COVID-19 reported in New York City as of May 2nd, this suggests a very approximate infection fatality rate of around 0.8%, again, without accounting for downward bias because of censoring and upward bias because of delay to seroconversion, but in any case, basically in line with prior estimates. Next slide. This is a research letter from a group in Los Angeles County that was actually published today in JAMA, where they recruited participants from a market firm's proprietary database that is supposedly representative of the county, and invited a random group of 1,952 participants for antibody testing via lateral flow immunoassay. 51% of those invited ended up being tested, and you can see some of the characteristics of those tested with the left column showing the entire sample size and the right showing characteristics among the 35 who tested positive. And after weighting by sex, race, ethnicity, income, and test characteristics, their findings suggest a prevalence of between around 2.5% to 7%. And the authors suggest, based on this, that there are about 43 times more infections than documented cases. However, the identification of their sampling population by this marketing firm is quite opaque, and with limited response rate, the study appears highly vulnerable to selection bias, with unknown effect on the findings here. There have also been some specific concerns about the performance characteristics of the assay used in this study, which was the same LFIA used in the Santa Clara study. Next slide. Here we see preliminary results announced May 16th from a representative community serosurvey of 4 zip codes near Boston, another area of relatively high prevalence. 5,000 residents were invited via email or flyer to participate, of whom 1,000 expressed interest and 786 were ultimately deemed eligible by being currently asymptomatic with no history of previously positive COVID-2 testing. 750 people ultimately participated and were tested for both antibodies and nasopharyngeal PCR. The investigators found an overall seroprevalence of around 10%, ranging from 6.3% to 13.3%. They also found that 2.6% of people were asymptomatically PCR positive. Again, a very rough estimate, but as in New York State, this suggests that there have been about 10 times more infections than identified cases, and that there is a very approximate infection fatality rate of around 0.8%. Next slide. And now on to what is perhaps the most methodologically rigorous large-scale serosurvey to date, the first round of a nationwide serosurvey in Spain called ENA-COVID, with initial results announced on May 13th. The report linked below describes a nationwide representative sample of 60,983 participants recruited from April 27th through May 11th, with a participation rate of 62.4%. They used a combination of rapid lateral flow immunoassays in all participants, as well as ELISA testing for the 89% who agreed to give a blood sample. The LFIA is the Orient Gene Biotech test, for which the manufacturer reports a sensitivity of 97% and a specificity close to a 100% for IgG. And in validation testing, the investigators found a sensitivity of 79% and specificity also close to 100%. Agreement between the rapid test and the ELISA was 97.3%. So in this study, they found an estimated prevalence of COVID-2 IgG of 5%, equivalent to about 2.35 million people, with a 95% confidence interval from 4.7 to 5.4%. They found that 83% of those who reported having had a positive PCR had a positive IgG, and that among those who did not report any symptoms, 2.5% had positive IgG. There's marked geographic variation, as you can see on the map, with the areas around Madrid exceeding 10% and other areas less than 2%. You may begin to recognize a pattern, as this suggests that there is an approximate ratio of 1 reported case for every 10 infections. With 27,100 known deaths from COVID-19, there is in Spain, again, a very rough estimate of a 1.2% infection fatality risk. Next slide. Now for a different approach to estimating prevalence. This was a modeling study published in Science on May 15th that jointly analyzed French hospitalization data, which is likely to be less biased than reported cases, with the results of the detailed outbreak investigation aboard the Diamond Princess cruise ship, where all passengers were tested. They do this in an attempt to disentangle the risk of hospitalization from the probability of infection, and then use mechanistic compartmental modeling to estimate transmission dynamics within France. Using this approach, they find that 3.6% of infected individuals are hospitalized overall, with the lowest rate of 0.2% in females under 20 and the highest rate of 45.9% in males over 80, and an infection fatality rate of 0.7%, fairly consistent with other estimates we've discussed previously. Their model also suggests that the national lockdown in France resulted in a 77% reduction in transmission, as shown in the top left figure, with the reproductive number falling to an estimated 0.67. Now, as shown in the figure in the bottom left, their findings suggest that by May 11th, between 1.8 and 4.7 million people will have been infected in France, with the uncertainty derived from the probability of hospitalization given infection. And that represents between 2.8 and 7.2% of the population. As shown in the figure on the right, the regional estimates suggest the highest proportion infected in the area around Paris at 9.9%. Their estimates are supported by reported seroprevalence of 3% among blood donors in certain areas of France, which is consistent with their model predictions for this population after accounting for a 10-day delay for seroconversion. Next slide. There are several issues with considering only reported, confirmed, or probable COVID-19 associated deaths, as highlighted by this paper from the New York City Department of Health, published in MMWR on May 11th. Reported deaths may underestimate deaths attributable to the pandemic by not including deaths from COVID-19 in the absence of diagnostic testing or with false negative testing, deaths outside of the healthcare setting, or deaths from COVID-19 that were not suspected to be related to COVID-19 by a healthcare provider. Reported deaths also do not count deaths that are not directly associated with SARS-CoV-2, but which result from other consequences of the pandemic, like changes in healthcare access, socioeconomic or psychological stresses, or other causes. And so to estimate deaths attributable to the pandemic, these investigators looked at excess mortality from March 11th to May 2nd, above expected seasonal baseline levels based on the prior 4 years. The figure shows time on the x-axis and death on the y-axis, with the dark blue being confirmed COVID-19 deaths, the light blue being probable COVID-19 deaths, and the black line being an estimate of all-cause excess death. They found that there were 24,172 excess deaths during the time period, 57% of which were laboratory confirmed, and 21% of which were probable COVID-19 associated deaths. This left 5,293 additional excess deaths, or 22% of the total. Excess deaths may thus be an important measure to follow over time that is more inclusive to the full impact of the pandemic. Next slide. Along these lines, thinking about the inclusive effects of the pandemic is the case of Sweden, which has taken a relatively unique approach to the pandemic. In Sweden, gatherings larger than 50 people have been banned and high schools were closed, but businesses have been generally allowed to remain open with minimal formal restrictions, aside from suggested physical distancing and working from home when possible, with high-risk populations like the elderly told to stay home. There have been some criticisms of this approach, prompting this editorial reference in The Lancet on the left, published in early May by the former state epidemiologist of Sweden, where he basically argued that there was not much point in trying to control the epidemic because everyone was going to get this infection over time, and that focus was better served on protecting the vulnerable. He also quoted non-specifically that around 25% of Stockholm had already been infected, although did not have any specific evidence for that per se. In any case, there's been a lot of interest as to how their approach might work in comparison to other areas with more strict and formal social distancing measures. Interestingly, Sweden has also been much more lax about testing and tracking the spread of SARS-CoV-2, which has made comparisons somewhat challenging. But there have been 2 preprints posted this week that help to shed some light on the impact in Sweden. The first we wanted to highlight by Moding and colleagues is posted on medRxiv on May 15th. They use Swedish register data of total weekly deaths and total population at risk to estimate age and sex-specific weekly death rates for 2020 and the 5 previous years, focusing on people over the age of 50. They found a substantial rise in excess mortality starting in the first week of April, about a month after the epidemic in Sweden began, with more pronounced rises in particular among the older age groups. This is illustrated in the central figures, which are stratified by weeks 11 through 16, with the x-axis showing age groups from 50 and up, and the y-axis showing the rate ratio of death rate for men relative to the prior 5 years. You can see as the weeks progress along that the rate ratio rises over time until by week 15 and 16, the inclusive measure of excess mortality is substantially higher in virtually every age group. The rightmost figure, however, concerns the potential economic upside of less restrictive measures. This comes from the second preprint, which was posted by Anderson and colleagues on May 12th, and compared aggregate consumer spending between 860,000 active customers of a large Scandinavian bank in Sweden and Denmark, where the epidemic began at the same time as Sweden, but substantial formal restrictions in social and economic activities were put into place. Denmark is on the top and Sweden is on the bottom, and the x-axis is the date. The y-axis is daily spending as a percentage of average spending in 2019, with the red line representing spending in 2020 and the gray line 2019. You can see the dashed gray line vertical there kind of midway through, where the shutdown in Denmark began, and the divergence between 2019 and 2020 spending thereafter. They found that aggregate spending dropped by about 25% in Sweden and 29% in Denmark, suggesting that while more limited measures in Sweden did lead to more economic activity, most of the economic decline was independent of these formal measures. The final word on Sweden's strategy remains to be seen, but early evidence thus suggests substantial increases in mortality with little gained in terms of economic protection. Next slide. So in sum, here are some key points about population infection estimates. As we've discussed, we are starting to see results from the first higher quality serosurveys that have more rigorous sampling methods. While reported cases do indeed represent the tip of the iceberg is perhaps not quite as big as we might have hoped, perhaps only about 1 order of magnitude difference. As a result, the current population estimates continue to suggest that this infection is quite severe on a population level with a greater than 0.5% infection fatality rate. Finally, excess deaths above expected seasonal levels may represent a more inclusive measure of the impact of the pandemic.
Okay, so there is some incredible and impressive science that's revealing the details of the origin story of the SARS-CoV-2 virus. And so we're going to try to go through that carefully next. On February 3rd, Nature published a remarkable article describing the identification of SARS-CoV-2 in Wuhan. So researchers at the Wuhan Institute of Virology took samples from 7 patients admitted at the time to the intensive care unit from the initial outbreak. 6 of these patients were sellers or delivery men at the now famous seafood market in Wuhan. Since the context was similar to how SARS started, they used pan-coronavirus primers to test the samples. They found that 5 of the samples were positive for coronaviruses. They then used next-generation sequencing and generated over 10 million reads, and then filtered out the human genome, and were left with nearly 1,600 reads. Of these, around 87% matched to sequences of SARS-like coronaviruses. They then assembled the genome and found it had 79.6% sequence identity to SARS-CoV. In the figure here, you can see the phylogenetic tree that they generated for some of the alpha and beta coronaviruses. The blue arrows here and here point to MERS-CoV and SARS-CoV, and the green arrows point to some of the common human coronaviruses. The viruses highlighted in red here are the 5 SARS-CoV viruses that they had isolated from those first patients in Wuhan. As you can see here, there's a known bat coronavirus called RATG13 that was the closest relative to SARS-CoV with around 96% sequence identity. In the figure here, you can see the percentage homology across the viral genome. So the viral genome is here. 100% homology would be identical to SARS-CoV-2. So the blue line is the bat coronavirus RATG13 that I just mentioned, and as you can see, it's quite similar to SARS-CoV-2. The other lines represent other SARS-like coronaviruses, including SARS and some other bat coronaviruses. They quickly made their sequences publicly available and developed a PCR assay. On January 29th, The Lancet published a paper describing how researchers in China analyzed the viruses of 9 patients in Wuhan from 3 different hospitals very early on. Some of the characteristics about the patients are shown in the table here, including that 8 of them had exposure to the seafood market. They generated 8 complete genomes, as you can see on the right here. And they found overall that there was a greater than 99.98% sequence identity between these viruses. So they note that this degree of sequence identity suggests an extremely recent emergence of the virus. They also note that, that their data are consistent with a bat reservoir, but make the case for an intermediate host. Among the evidence for their claim, they note that the outbreak was first reported in December 2019, when most species of bat in Wuhan are hibernating. They also note that no bats were sold or were reportedly found at the seafood market, whereas multiple other animals were sold there. On March 17th, Nature published a fascinating letter looking further at the origins of SARS-CoV-2. You can see the SARS-CoV-2, a schematic for the SARS-CoV-2 genome at the top here, and then they've blown up the spike, the S protein here, and you can see the S1 subunit and the S2 subunit here and here. On the bottom of the slide, you can see a close-up of the receptor binding domain, and what you can see is a comparison of the various amino acid sequences for 6 different SARS-like coronaviruses. So first is SARS-CoV-2, the bat virus that we talked about, RATG13, a pangolin coronavirus, SARS, and 2 other SARS-like coronaviruses. Up here, you can see an expanded region for the S1, S2 cleavage site for the viruses. And the authors make a few very important points. First of all, they note in general, the receptor binding domain is the most variable part of the coronavirus genome. Second, they note that the RBD, the receptor binding domain for SARS-CoV-2, is quite distinct from that of SARS-CoV. While both are optimized for binding to human ACE2, they do so in different ways. For instance, 5 out of these 6 key residues that they've highlighted here are different between the 2 viruses. This is strong evidence that the virus was not a product of a laboratory studying SARS-CoV. If it were, it would have had an RBD much more closely related to SARS-CoV. Third, the authors note the importance of an insertion of a polybasic cleavage site, which you can see here with the red arrow highlighting the area between S1 and S2. They note there's evolutionary precedence for selection of polybasic cleavage sites in influenza. These sequences increase pathogenicity and enhance cellular entry since they are easily recognized by certain cellular proteases. Recall that cleavage of the S1 and S2 subunits is an important priming step for cell entry. Finally, the authors note that this insertion sequence is distinct from all of the other known related SARS-like coronaviruses to date in this area, and what they predict that other similar coronaviruses with partial or complete polybasic cleavage sites will be discovered. In another paper published in Nature on March 26th, scientists analyzed sequences of coronaviruses identified in pangolins. So let's walk through the figure here on the left. So this is an amino acid, this is the map of the amino acid sequences for the receptor binding domain of the S protein for 6 different SARS-like coronaviruses, including from top to bottom, SARS-CoV, SARS-CoV-2, a Guangdong pangolin coronavirus, and the bat coronavirus, RATG13 that we just discussed, along with 2 other coronaviruses. I've added red arrows to highlight 5 critical amino acid residues for ACE2 binding, which you can see here at the bottom of the slide. The RBD of the Guangdong pangolin coronavirus has 97.4% sequence identity with the RBD of SARS-CoV-2. It has the closest homology to the RBD of SARS-CoV-2 of any known SARS-like coronavirus. And it shares 5 out of 5 of these key residues. The bat virus RATG13 only shares 1 out of the 5 residues. On the right, you can see the phylogenetic tree that they created, just if you look at the RBD for the coronaviruses. Again, showing that the Guangdong pangolin coronavirus and SARS-CoV-2 are more closely related at the RBD than SARS-CoV-2 is to the bat coronavirus RATG13. However, the overall genome, the bat coronavirus RATG is more closely related to SARS-CoV-2 compared with the overall genome of the Guangdong pangolin coronavirus. So they proposed two possible mechanisms to explain this conflicting cluster formation. So the first is a recombination event between a bat coronavirus and a pangolin coronavirus in pangolins, with subsequent transmission to a human. In this way, a closely related bat coronavirus like RATG13 could pick up a pangolin coronavirus RBD, and the result would be a virus with even more sequence identity to SARS-CoV-2. The second possibility is called convergent coevolution, whereby the RBD of a bat coronavirus was selected to be similar to SARS-CoV-2, perhaps also in pangolins. The authors note that the ACE2 receptor of pangolins and humans has 85% sequence identity, while that of bats and humans has 81% identity. Therefore, when bat coronaviruses pass into pangolins, those that can bind more easily to pangolin ACE2 will have a selective advantage. Because of the greater similarity between ACE2 and pangolins and humans, natural selection in the pangolins might also result in better ACE2 binding in humans. These are both intriguing possibilities to consider. On April 6th, Current Biology published another important article from researchers in China on this subject. As background, they note that a SARS-like coronavirus was reported in 2 dead pangolins from the Guangdong Wildlife Rescue Center in late October 2019. They analyzed the genomes of the virus in these pangolins and found that they had 80 to 88% sequence identity to known SARS-like coronaviruses. Given the timing around the likely start of the COVID-19 outbreak, they hypothesized that this new coronavirus could be closely related to SARS-CoV-2. They then assembled the genome and this new virus they called Pangolin-CoV. Like other coronaviruses, it contains 6 major open reading frames and 4 accessory genes. Here, what you can see is a schematic with the genome for SARS-CoV-2 on top, the Pangolin-CoV, and the bat coronavirus RATG13 that we talked about on the bottom. And you can see the percent similarity between the various parts of the genome here, where the first number, for instance, for the S, is the nucleotide similarity, and the second number is the amino acid, identity. So overall, they found that the genome of pangolin coronavirus showed 90.6% sequence identity to RATG13 and 91% sequence identity to SARS-CoV-2. There was very high S protein amino acid identity between Pangolin-CoV and SARS-CoV-2, as you can see here, 97.5% amino acid sequence identity between the two viruses. Based on all of their analyses, they then estimated phylogenetic trees based on the nucleotide sequences of the whole genome, and then based on the sequences for specific proteins. In all cases, Pangolin-CoV, bat-CoV RATG13, and these new SARS-CoV-2, newly discovered SARS-CoV-2 viruses all clustered together. And they called this group the SARS-CoV-2 group. And you can see that here represented in the phylogeny on the right. They proposed that the Pangolin-CoV could be a potential common ancestor for the bat coronavirus and SARS-CoV-2. In the same paper from the last slide, the authors note that prior recombination events in the beta coronavirus group complicate generating an accurate phylogeny. These events may occur in an intermediate host when 2 distinct coronavirus genomes can recombine. An example of a place where this is likely to have happened is in the S protein of SARS-CoV-2. The S1 subunit contains the RBD that consists of about 193 amino acid residues. The S1 of Pangolin-CoV is more closely related to that of SARS-CoV-2 than to that of RATG13. For instance, within the RBD, there's only 1 amino acid difference and not one of the 5 critical ACE2 binding residues between the SARS-CoV-2 and the pangolin coronavirus. In comparison, RATG13 has 17 amino acid changes, including 4 in key ACE2 binding locations. Besides the RBD, the S1, S2 cleavage site is also important for transmissibility and pathogenicity of SARS-like coronaviruses. Again, this is that important priming step for cell entry that we've discussed. So the authors again note that the importance of this insertion here that you can see on the slide of this 4 amino acid sequence in the region of the S1, S2 cleavage site for SARS-CoV-2. You can see there are 5 SARS-CoV-2 genomes here. They all have this sequence, but none of the other closely related SARS-like coronaviruses, including RATG13, Pangolin-CoV, SARS-CoV, or other bat SARS-like coronaviruses have that sequence. As we mentioned before, with the addition of this sequence, SARS-CoV-2 contains what is called a polybasic cleavage site, and that can also be called a putative furin recognition motif. This is similar to what MERS-CoV has, but again, none of these more closely related SARS-like coronaviruses have this sequence. A putative furin recognition motif is essentially an amino acid sequence that is easily recognized and cleaved by particular cellular proteases. Since cleavage of S1 and S2 is a key priming step for cell entry, the authors theorize that the presence of this sequence might facilitate viral entry into host cells. This again is evidence for insertion or recombination events between viruses. On May 7th, Nature published another important paper in this area. These scientists examined samples from 25 Malayan pangolins from a rescue center between March and August 2019. Surprisingly, they found that 17 of these pangolins were infected with a SARS-like coronavirus. Using PCR primers from highly conserved regions, the pangolins who tested the pangolins who actually tested positive developed a clinical illness that included shortness of breath, lack of appetite, and crying. 14 of the 17 pangolins died within about 6 weeks of each other. An analysis of the lung tissue after their death showed diffuse alveolar damage and other pathology. They determined it was a single virus that had infected all of these animals. They assembled the genome, and they found that there was high sequence identity between the S, E, M, and M genes of this pangolin coronavirus with SARS-CoV-2 and also with other SARS-like coronaviruses, as you can see in this table. So here you see first nucleotide and then amino acid identity between this pangolin coronavirus, SARS-CoV-2, SARS-CoV, the RATG13, and 2 other bat SARS-like coronaviruses. And again, quite impressive similarity here. What the authors noted is that there was differential similarity between these viruses at various regions of the S gene, and you can see that represented here. So at various regions, for instance, at this region, the bat coronavirus RATG13 and SARS-CoV-2 were more closely related, and Pangolin-CoV seemed to be more distantly related. Whereas the cluster was different in this region, and also different further along in this region. So again, they note that this is called conflicting cluster formation, and again, it's a strong indication of recombination events. They note that the pangolin coronavirus that they identified is related to SARS-CoV-2, but it's unlikely to be directly linked to the outbreak because of its substantial sequence differences outside of the RBD with SARS-CoV-2. They also importantly note that since the Malayan pangolins appeared to have died from this viral infection, they're unlikely to be native hosts and rather more likely to be an intermediate host. They further note that since pangolins and bats have overlapping ecological niches, both are nocturnal and eat insects, they are likely to have close contact in the wild and are therefore well suited to be, and therefore pangolins are well suited to be potential intermediate hosts. Next, on May 11th, a fascinating study was published in Current Biology. In this study, researchers from China and Australia collected a variety of bat samples between May and October of 2019. They pooled the samples together and then used metagenomic next-generation sequencing to map the sequences onto a previously characterized SARS-like bat coronavirus and to SARS-CoV-2. They generated two consensus sequences, which they called RMYN01 and RMYN02. They then went back and looked for the consensus viruses among the original bat samples, and they actually found RMYN02 in one bat sample. Overall, they found that this virus had 93.3% sequence identity between SARS-CoV-2 and this new virus, again, RMYN02. It was least similar in the S gene, where there was only 62.4% homology in the receptor binding domain. Based on the sequence in the RBD, they expect decreased or potentially no binding ability to ACE2. Importantly, however, they found a partial polybasic cleavage site insertion in this new virus, which is represented here on the slide. And again, just to go through this slide, so these are amino acid sequences for SARS-CoV-2, this new RMYN02 virus, the RATG13 virus, and several other SARS-like coronaviruses, including near here, close to the bottom, the original SARS coronavirus. So going back here, you can see this insertion of three residues a partial insertion of a polybasic cleavage site. And importantly, this is exactly what Anderson and colleagues had predicted in their letter to Nature that we discussed, that was published nearly 2 months before this was published back on March 17th. Again, further evidence for the natural origin of these viruses. It's important to understand how SARS-like coronaviruses interact with their natural reservoir host, which is presumed to be bats. In a fascinating study published in Scientific Reports on April 29th, authors showed that MERS-CoV can persistently infect bat cell lines. They note that mathematical models have proposed a persistent infection model with low levels of periodic shedding as how these coronaviruses exist in the bat reservoir. As shown here, these scientists inoculated bat cells with MERS-CoV, as you can see on the left. Many of the cells died, but some survived. By around day 42, they found persistent low levels of MERS-CoV in the surviving bat cells, along with increased levels of interferon. They analyzed the MERS-CoV in the surviving cells, which you can see represented on the right side of the slide here. And what they found was that these MERS-CoV viruses had a deletion in one of the accessory genes called ORF5. They determined that this was selected for when the MERS-CoV was passed through the cells. When they re-challenged the cells with wild-type MERS-CoV, they were resistant to superinfection, possibly because of the increased interferon expression pattern. They also found that downregulating certain transcription factors, like interferon regulatory factor 3, led to increased MERS-CoV transcription and increased cell death. This model shows the delicate balance between the cytolytic viral and host defense factors that allows for persistent infection. It also suggests, with the addition of certain stressors that might lead to changes in cellular gene expression, there can be increased coronavirus expression and therefore probable shedding. We think the point about persistence of infection in some reservoirs is incredibly important because there's now good evidence that SARS-CoV-2 can infect both cats and dogs. There's evidence that cats can infect each other, but not yet evidence that dogs can propagate the infection. If either of these organisms, or potentially if other animal reservoirs, had persistent infection with SARS-CoV-2, it would be important for us to know, since that could be a potential ongoing source of infection. Along these lines, there is a coronavirus called feline coronavirus, which is an alpha coronavirus that can actually cause persistent infection in cats. In the study shown here, one cat shed the virus for 7 months after the cat was isolated from other cats, excluding the possibility of accidental reinfection with the same virus. The figures here show immunohistochemical staining in the ileum and the colon with the virus in these chronically infected cells, in again, the ileum and the colon. Before concluding this section, we want to take 1 minute to discuss an update in viral tropism and cellular entry, which after all is very closely related to the S protein that we've just talked so much about. In past updates, we've discussed in detail evidence for extrapulmonary infection. We wanted to highlight this autopsy study from May 13th that was published in the New England Journal. This series included 27 patients, and they performed quantitative RNA PCR for SARS-CoV-2 from multiple tissues. The most important part of the figure is on the left here, where you can see a gradient of SARS-CoV-2 copies per cell, with white being no copies, gray being more, blue being even more, and red being the most. The highest levels of SARS-CoV-2 copies per cell were detected in the respiratory tract, and lower levels were detected in the kidneys, liver, heart, brain, and blood. In a past update, we showed evidence for SARS-CoV-2 entry into T lymphocytes, which have extremely low levels of ACE2 expression. And the autopsy study from the last slide shows viral infection in brain and liver, which also have low levels of ACE2. There's been a lot of speculation that ACE2 may not be the whole story when it comes to cell entry for this virus. We wanted to highlight an interesting and hypothesis-generating study published in iScience on May 13th. This paper suggests DPP4, which is the entry receptor for MERS-CoV, may be another possible entry receptor for SARS-CoV-2. They use bioinformatics approaches to predict the interactions between human proteins and the spike proteins of MERS-CoV, SARS-CoV, and SARS-CoV-2. They found that ACE2 and DPP4 were both expected to have favorable interactions with SARS-CoV-2, as shown here on the left of the slide, where favorable interactions have more negative free energy. ACE2 interactions are in purple, and DPP4 interactions are in green. They note that the interaction with ACE2 for SARS-CoV-2 is more favorable, so there might be priority for ACE2 binding, but they do predict ability of DPP4 binding. In contrast, as you can see, MERS-CoV exclusively interacts with, is exclusively predicted to interact with DPP4, and SARS-CoV only with ACE2. The schematic on the right shows the model for cell entry they propose, including that SARS-CoV-2 may bind both important entry receptors. This might explain the tropism of the virus for cells with non-ACE2 expression. We hope to see more studies about this, including in vitro studies to explore this intriguing hypothesis. These are some important points to conclude from the studies that we just went through. First, there's strong evidence for the natural origin of SARS-CoV-2, and given the numerous similar viruses circulating in bat and other animal reservoirs, we need to be prepared for events like this to happen again. Second, bats are a probable natural reservoir, and pangolins may be an intermediate host for SARS-CoV-2. Third, an in vitro model describes persistent MERS-CoV infection of bat cells. Cats and dogs can be infected, and it will be essential to determine whether other animal reservoirs can develop persistent infections. Finally, as we have shown previously, the virus has extensive extrapulmonary tropism. There's an intriguing new bioinformatics work suggesting that SARS-CoV-2 may be able to use the DPP4 entry receptor in addition to ACE2, and this will certainly need to be worked out further. We want to spend a few minutes now on antibodies, immunity, and vaccines. So we intend to spend more time discussing antibodies and immunity another time, and this is clearly an area where there continue to be major gaps in our knowledge, but we did want to discuss two recent important papers. The first is this recent preprint posted to medRxiv on May 5th from authors at Mount Sinai in New York City, and this characterizes humoral responses in a large cohort from New York City. They characterize the antibody dynamics of a group of 1,343 suspected or confirmed symptomatic patients with COVID-19. Of the patients, 624 were confirmed positive by PCR. Of these, only 3 did not develop antibodies, and those 3 were self-reported PCR positive. Interestingly, the authors note that IgG antibodies develop over a period of 7 to 50 days after the onset of symptoms, with a median of 24 days. We think this is important because it's longer than what was previously reported in a study published in CID, whose reference is also on this slide. That study reported a median of 14 days. The authors of this New York City-based preprint found that higher antibody titers were associated with longer symptom duration and longer time from the onset of symptoms, as you can see in the slide here. We also want to highlight this very elegant study published in Cell on May 14th. In the study, the scientists characterized the adaptive immune responses to SARS-CoV-2 in recovered patients. Importantly, they characterize the various antibodies and distribution of reactive T cells to different viral proteins in recovered patients. All of this work may be helpful in the design of effective vaccines, and particularly in effective vaccines that mimic the natural adaptive immune response to the virus. One exciting finding, which is consistent with what we presented last time from the preprint by Braun and colleagues referenced on this slide, is that some individuals without prior SARS-CoV-2 infection also have SARS-CoV-2 reactive T cells. In the current study, the authors found around 50% of their historical healthy donor cohort, historical healthy donor cohort had such cells, which were more likely to be reactive to non-spike proteins. And you can see that here, where the COVID patients are, the patients who are recovering from COVID are in blue, and the unexposed individuals are in black. The dashed line here, if you're above that line, that indicates that you have reactive cells. So this is suggestive evidence that cross-immunity, perhaps from the common human coronaviruses, may at least partially explain the disease severity spectrum seen for COVID-19. If this is true, it also potentially means that a vaccine that could provide even some protection could have a massive positive impact on overall disease severity. In terms of vaccines, we wanted to give a quick update on this topic. It seems clear that an effective vaccine against SARS-CoV-2 is an important step towards getting out of the current crisis we're in. An exciting study was released in Science on May 6th, when researchers from China reported an update on an inactivated vaccine they're developing. They call their vaccine PicoVax, for Purified Inactivated Coronavirus Vaccine. The researchers isolated SARS-CoV-2 strains from 11 patients. They grew and purified the virus in cell cultures and inactivated it. They first inoculated mice with the vaccine on day 0 and day 7, with the vaccine product along with an alum adjuvant, and they inoculated a comparison group with a saline sham. They then measured specific amounts of IgG to S, RBD, and N protein. And you can see on the figure on the left here that both IgG levels and neutralizing antibody levels increase over a period of about 6 weeks. They then tested the vaccine in rhesus macaques. So they gave the macaques a 3-dose series. They measured antibodies similarly to how they did with the mice. And then they inoculated the macaques with SARS-CoV-2 through an intratracheal injection and compared the macaques and had a comparison group. Placebo-treated macaques, so macaques who had been vaccinated with a placebo, developed viral shedding and pneumonia. However, all vaccinated macaques were largely protected. All 4 monkeys that received a high-dose vaccination had no detectable viral load in the pharynx. There was no evidence of antibody-mediated enhancement. However, they did not measure direct T cell responses in the monkeys to the vaccine. On May 18th, so actually earlier today, the Cambridge-based company Moderna issued a press release describing the results of their phase 1 trial for a candidate vaccine they call mRNA-1273. They report that it elicited high neutralizing antibody titers in the 8 participants who enrolled. We'll of course await to see full details in a peer-reviewed format, but this again is exciting news. These are some key summary points from the last 3 slides, including that antibody dynamics are still being worked out. Some individuals may have cross-protective cellular immunity, perhaps from prior infection with some common human coronaviruses. And finally, that there's promising work being done on vaccines.
Okay, so next we want to try to answer with the best available evidence a question that's coming up more and more, and that is, after a patient has already been diagnosed with COVID-19, what is the role for repeating PCR later in disease? Next slide. And the reason this comes up is because many institutions, hospitals, skilled nursing facilities, rehabs, outpatient labs, workplaces have been using PCR as a sort of test of cure prior to getting rid of isolation and transmission-based precautions. Here's an example of the test-based strategy recommended by the CDC, whereby someone can be cleared from precautions after resolution of fever, improvement of respiratory symptoms, and 2 negative PCR tests collected greater than 24 hours apart. Does this make sense? Next slide. With PCR testing later in disease or after recovery, we also end up with alarming and confusing headlines and tweets like these. Recovered coronavirus patients are testing positive again. Next slide. So to get at this larger question, we're going to go through the evidence or lack thereof supporting answers to the following 4 questions. Next slide. So first, because in these cases we are essentially using viral RNA as a proxy for transmission risk, the first question is how well does identification of viral RNA correlate with culturable, i.e. infectious virus? And first we'll return to this important Wolfel study on the left, which was published in Nature of 9 mild cases of COVID-19, where they isolated live virus from samples up to day 8 of infection, but none thereafter, as shown in the left side of the figure on the left. And while culturable virus did correlate with quantitative viral load, as seen in the right half of the figure, viral RNA remained intermittently positive for 1 to 2 weeks longer. The second study to help us answer this question is this study on the right of a nursing home outbreak published earlier this month in the New England Journal, where they performed universal PCR testing with viral culture at 2 time points 7 days apart. In the figure, you can see results of PCR testing and culture, with the x-axis representing days before or after symptom onset, the y-axis representing cycle threshold values, and the red dots are those samples with positive viral culture. You can see the density of positive viral cultures in the pre-symptomatic phase, a bit less in the early phase, and even less about 7 to 10 days after symptoms. Importantly, these patients had disease across the clinical spectrum, and a substantial percentage actually died. And so there was a fair bit of severe disease here. The evidence, thus, is limited, but suggests that a positive PCR correlates with infectious virus early in disease, but in later disease, much less so, if at all. Next slide. The next question is how well does PCR positivity correlate with disease severity and timing after symptom onset? The figure on the left comes from a study published in Nature on April 15th of 414 throat swabs from 94 patients. So not nasopharyngeal swabs, but we'll kind of get the point. The curves at best fit here suggest that PCR positivity gradually decreases towards the detection limit at day 21, and that there's no difference after stratification by disease severity. What I want you to pay attention to are the individual patient lines here, which frankly are all over the place. They're positive, negative, and then positive again. Look, for example, at the patient who becomes negative before becoming positive again, all the way at day 28. This certainly fits with the clinical experience of anyone who's taking care of these patients with this disease with repeated testing. The right figure is from a recent paper in Emerging Infectious Diseases, modeling the length of PCR positivity based on a number of hospitalized cases. The top figure is of mild cases and the bottom figure of severe cases. And their findings suggest that only 50% of nasopharyngeal swabs were clear by day 22, and that this was longer for severe cases. So these data and other papers suggest that in individual patients, there's not really a predictable and steady extinction in viral load, with perhaps some of this related to sampling variation. Importantly, prolonged RNA shedding is common in both mild and severe cases. Next slide. Which brings us to the third and perhaps most important question. Despite this prolonged isolation of viral RNA in some patients, when during the COVID-19 disease course does transmission occur? To answer this, I will highlight figures from 2 studies we previously discussed. The first is this JAMA Internal Medicine paper on the left, with the figure describing a detailed contact tracing response to 100 cases in Taiwan. In the figure, the number of contacts is the blue bars, which corresponds to the left y-axis, and the clinical attack rate is in the light blue points corresponding to the right y-axis, based on different categories of days from symptom onset to first exposure, which is down on the x-axis. So the important point from this figure is that, if you look at the light blue line, that they found no secondary transmissions from contact exposures after the 5th day of symptom onset, suggesting a relatively short infectious period. At the very least, this suggests that late transmissions are at least rare. The figure on the right comes again from this Nature paper by He and colleagues, in an analysis where they evaluated 77 detailed transmission pairs, and based on the serial interval, i.e. the amount of time between symptom onset in the transmitter and the symptom onset in the transmittee, estimated the infectiousness period of SARS-CoV-2. And so after peaking in the pre-symptomatic period, you see a rapid and substantial decline towards 0 by day 7 or so. So together, all of this suggesting that transmissions greater than 5 days after symptom onset are rare, if they occur at all. Next slide. So then bringing all of that together, how helpful is repeat PCR likely to be after COVID-19 is diagnosed? Okay, so go ahead and click. All right, so given first that viral RNA does not seem to correlate with infectious virus after day 8 after symptom onset. Second, that prolonged and intermittent viral shedding is frequently seen in both mild and severe cases. And third, that transmissions after 5 days of symptoms have not been documented and are likely to be rare. We suggest that PCR testing later in disease for clearance of transmission-based precautions is unlikely to be helpful and probably delays care, causes distress to patients and healthcare workers, and creates these false narratives of reinfections. The caveats to this, of course, are in critical cases and in the context of high-risk aerosolizing procedures, where less data are available and greater caution may be considered.
Okay, so we wanted to highlight a new syndrome that has gotten increasing attention over the last 2 weeks. It's being called pediatric multi-system inflammatory syndrome and has similarities to Kawasaki disease. On April 9th, clinicians at Stanford Children's Hospital reported a case of a 6-month-old child they diagnosed with Kawasaki disease and COVID-19 simultaneously. The patient responded to IVIG and high-dose aspirin. They noted that others should be on the lookout for additional cases. Since that time, centers from around the world have reported similar cases. Classic Kawasaki's includes fever and 4 to 5 additional criteria in the absence of an alternative diagnosis. The largest published series describing this new syndrome was published in The Lancet on May 13th. The authors reported 10 cases of a Kawasaki-like syndrome at their hospital in the Lombardy region of Italy during the local COVID-19 outbreak from February 18th to April 20th. Of the cases, 5 fit the definition for complete Kawasaki and 5 were incomplete. You can see some of the patient characteristics here in this table. In particular, 4 of the patients actually met, 5 of the patients also met criteria for macrophage activation syndrome. The authors looked back at the diagnoses of Kawasaki disease in the preceding 5 years from January 1st, 2015 through February 17th, 2020. And they found 19 patients during that time period who were diagnosed with KD, including 6 with incomplete disease. None of those cases also met criteria for macrophage activation syndrome. There's still some controversy about the incidence calculations that are reported in the paper, but overall it seems likely that this is a true increase over what would be expected given the data that they present from their previous 5 years. They also note that the average age of the previous cases was 3.0 years, while the average age of the new cohort was significantly older at 7.5 years with a p-value of 0.003. As you can see here, 8 out of 10 of the patients, so all highlighted in with red boxes, tested positive either by serology or PCR or both for SARS-CoV-2. In the last couple of weeks, there's been tremendous interest, as we mentioned, in this area. And last week, New York State Department of Health released an interim case definition, and that is shown on this slide. They have reported over 102 cases of this syndrome in New York State alone. Interestingly, in 2005, JID published a series of 8 patients with KD who had respiratory secretions positive for another human coronavirus at the time what that was called New Haven coronavirus. And they raised the possibility of an association between this infection and the inflammatory condition. The authors identified 54 children who received the diagnosis of Kawasaki disease in the preceding 3 years at their institution. They had respiratory samples for 11 of the patients. They matched each patient who had Kawasaki's with 2 controls, and those 2 controls were the first 2 children within 6 months of age of the subject who had specimens obtained within 1 week of the subject. What they found was that 8 of the 11 case subjects and just 1 of the 22 control subjects tested positive for this particular coronavirus. Taken together with the emerging data from our current pandemic, it seems clear that this is an important area that we'll be hearing a lot more about in the days ahead.
All right, so next, just a few quick points about something that has been getting a lot of discussion, BCG vaccination. So BCG is a vaccine with low to moderate efficacy in preventing tuberculosis, but is also thought to perhaps have other nonspecific benefits, with some randomized control trials suggesting that BCG reduces rates of other respiratory infections and even mortality in children. The mechanism is thought to involve antigen-independent activation of B and T cells in what has been termed heterologous immunity, as well as potentially enhancement of innate immunity through epigenetic reprogramming in what has been called trained immunity. This has led some to wonder whether BCG may be protective in general with COVID-19 and contribute in part to the lower severity of illness in children. This interest was further fueled by an ecological study posted as a preprint to medRxiv on March 28th, with the representative figure on the left suggesting that countries without universal BCG vaccination policies were being more severely affected by COVID-19. These data are weak, with many, many limitations, but for better or worse have inspired multiple clinical trials for the use of BCG in the context of COVID-19. Since then, however, we now have somewhat better observational data that is patient level rather than ecological in a research letter published in JAMA on May 13th. So this study comes from Israel, where BCG was routinely administered to all newborns from 1955 to 1982 with greater than 90% coverage, but only administered to immigrants from countries with high TB prevalence since 1982. So they looked at over 3,000 SARS-CoV-2 test results from individuals born in the 3 years before the change, as indicated in the left column, and just under 3,000 from individuals born in the 3 years after the change in the right column. And they found no difference in the proportion of positive tests in these groups. So, we'll have to wait to see the results from the randomized control trials, but for now, the best evidence does not support the hypothesis that childhood BCG vaccination later protects against COVID-19. Next slide. So as the pandemic carries on, we're starting to see some large-scale observations made around real-time monitoring for the prediction of disease. Next slide. In this paper published in Science on May 5th, a group calling themselves the Coronavirus Pandemic Epidemiology, or COPE Consortium, describes a collaboration between a number of well-known large cohorts that have joined together with the purpose of interrogating the pandemic through what they call the largest and most diverse patient population to date. As part of this, they developed a COVID symptom tracker mobile app that allows for self-report of data related to symptoms, exposures, and infections, with a workflow shown in this figure. Next slide. This app-based symptom tracker was first rolled out in the UK on March 24th, and then in the United States on March 29th. The first investigative application with this app is described in this paper published in Nature on May 11th that tried to answer the question of whether loss of smell or taste is truly a predictor of COVID-19 or not. So the app collects data from both asymptomatic and symptomatic individuals and tracks self-reported health information daily, including symptoms, hospitalizations, PCR testing, demographics, and pre-existing medical conditions. So this paper included a study period of March 24th to April 21st, during which nearly 2.5 million individuals in the UK and about 170,000 individuals in the US reported symptoms, with 32% indicating at least one potential symptom of COVID-19 during that time period. So nearly 16,000 users in the UK and 2,700 in the US reported having a PCR test. And in the UK cohort, they found that 65% of those who tested positive reported loss of smell and taste, compared to 23% of those who tested negative. After adjusting for age, sex, and BMI, after adjusting for age, sex, and BMI and a logistic regression and combining the UK and US data sets through meta-analysis, they found that users reporting a loss of taste and smell had an odds ratio of 6.74 of having a positive PCR test. They also evaluated for other queried symptoms in the UK cohort, as indicated in the figure in the gray boxes. And after adjustment for age, sex, and BMI, as well as for multiple testing, all were associated with a positive test, although with markedly lower odds ratios than anosmia, as you can see at the top. In the US, as indicated in the black boxes, only loss of smell and taste, fatigue, and skip meals were associated with a positive test result. Interestingly, they found an increasing odds ratio for the relationship between loss of smell and taste and a positive test over time, which they suggest is related perhaps to increased media reports and awareness of this as a potential symptom. They use a relationship between the queried symptoms and a positive test to build a predictive model, and then applied that model to the other 800,000 symptomatic individuals in their data set to suggest that approximately 140,000 were likely to be infected. This is a super interesting analysis suggesting the importance of loss of taste and smell, albeit among this self-selected group who might not be fully representative of the general public, as well as a proof of concept of identification of cases through mobile symptom trackers. Next slide.
We're all eagerly awaiting the remdesivir publications, which we expect will be out imminently. So we're not going to discuss, obviously, those now, but we did want to end with a brief treatment update. We wanted to highlight this one study, which is a treatment trial that was published in The Lancet on May 8th. The study was a multi-center open-label RCT at 6 hospitals in Hong Kong. Subjects were randomized 2-to-1 to receive a combination of lopinavir, ritonavir, ribavirin, and up to 3 doses of 8 million units of interferon beta-1 on alternate days versus lopinavir, ritonavir alone. Interferon was given early in the disease course out of concern that it might exacerbate the hyperinflammatory syndrome if given late. So again, wasn't given after 7 days of symptom onset. If patients presented within 2 days of symptoms, they received all 3 doses. If they presented at days 3 to 4, they received 2 doses. If they presented at days 5 to 6, they received just 1 dose. And again, if they presented on day 7 or later, they did not receive interferon. All interventions were started within 48 hours of hospitalization. The primary endpoint was time to negative NP swab for SARS-CoV-2. They ended up randomizing 86 patients to combination therapy and 41 to monotherapy with lopinavir, ritonavir only. On the top left here in this table, you can see some of the baseline characteristics of the patients in each group. The average time from symptom onset to start of treatment was 5 days in the combination group and 4 days in the control group, which means that the combination group, the median number of doses of interferon was 2. What they found was a significant difference in time to viral clearance from NP swab of 7 days for the combination group versus 12 days for the control group with a significant p-value of 0.001, as you can see in the table here, and also in the figure on the left here. They also found that the duration of hospital length of stay was decreased in the combination group at 9 days compared to 14.5 days, with a p-value of 0.016. When they analyzed the findings in the subgroups, the findings remained strong in the groups that were started within 7 days of symptoms, i.e. those who actually received interferon, and that's on the left here. Those who started treatment after 7 days of symptom onset, so that was 34 individuals in the combination group and 17 individuals in the control group, there was no difference across all of their endpoints. It's important to note that that the patients in this group that they enrolled they actually had quite mild COVID-19, and the deaths in each group were 0. So I think it's a little bit unclear how this can be extrapolated to patients with more severe disease. Additionally, there's an important point that we wanted to mention in the reference at the bottom of the slide in Annals, which is an important pharmacokinetic study of lopinavir ritonavir, and this was published on May 12th. This paper reports that levels of lopinavir ritonavir would need to be 60 to 120 times higher than are achieved at standard dosing of 400 milligrams with 100 milligrams twice daily to actually achieve inhibition of SARS-CoV-2. They argue that this data makes it seem unlikely that this medication will have any clinical use for COVID-19. The next several slides are updates of are updated lists of the major treatment trials that we have shown in the past. So on this slide, this hydroxychloroquine RCT is now published in BMJ. It was previously a preprint. This is the reference for the interferon study that we just discussed. This is our slide from last time with preliminary data from RCTs. Importantly, the French tocilizumab study that we discussed last time, there was a news report that the DSMB actually resigned after the announcement was made, and so we'll have to wait for hopefully some more clarification, perhaps in a peer-reviewed setting, to know what is going on there. We also updated the hydroxychloroquine trial tables. There are 2 additional large observational studies out of New York City that both showed no benefit. So to summarize some key points from today, There's mounting evidence for racial inequalities and outcomes for COVID-19. Multiple population-based surveys suggest about 10 infections per one reported case and an infection fatality risk greater than 0.5%. There's strong evidence for a natural origin of SARS-CoV-2 and high likelihood of future zoonotic events, so we need to be prepared. Bats are a possible, are a probable natural reservoir, and pangolins are a possible intermediate host. Antibody dynamics are being defined, but the time to development of IgG might be slightly longer than previously reported. Evidence does not support the use of SARS-CoV-2 PCR for clearance of transmission-based precautions, since PCR does not correlate with infectiousness after about 8 days of symptom onset. There's an emerging link between a pediatric inflammatory syndrome and COVID-19 that needs to be explored. It remains to be seen if learning from this will help us understand and treat the highly morbid hyperinflammatory phase that is seen in many adults with severe COVID-19. A large-scale real-time symptom monitoring study seems to confirm the importance of anosmia and ageusia in presentations of COVID-19. And finally, an open-label trial with interferon beta did show improvement in viral shedding, and it's the first major trial that has shown that. So we're going to end there, and thank you very much, and we'll see you again next time.